All right, good morning. Thank you for joining us for this week's Midwest Bioinformatics Showcase Seminar. Today's speaker is Laia Vancells Lopez, a PhD candidate in biomedical engineering at Purdue University, where she is advised by doctors Nan Kong and Leopold Green. Laia has a master's degree in data science from Wake Forest University, where she also did her undergraduate majoring in biochemistry. In addition to being an accomplished scholar, an awardee of things like the Premi Argot Award for Research Excellence from the Universitat Autònoma de Barcelona and the Wake Forest Excellence in Data Science Achievement Award, she's also an accomplished athlete with a D1 field hockey career. And she has combined these two interests through lots of different outreach programs. For example, with the Bridgeport Community STEM Access and Sports Engagement Program, the Orthopedic Research Society, or ORS, Open Doors STEM Access Initiative, and the MBA STEM Access Initiative for Underrepresented Youth Through Sports. Today, she will be talking about her work with agentic modeling. Her talk is titled Spatiotemporal Agent-Based Modeling of Immune Control Gates in the Tumor-Draining Lymph Node. And Laia, I will turn it over to you.
Thank you so much. And can you hear me all right? Perfect. Let me share screen. Okay. Thank you so much. So today I'm presenting the spatiotemporal agent-based modeling of immune control gates in the tumor-draining lymph node and how these give rise to what we identify as immune control gates. So before getting into the modeling, I just want to quickly ground us in the biology. So cancer isn't just uncontrolled growth, but it's also a system that actively adapts. It sustains its own proliferation, avoids cell death, rewires metabolism, and also it evades and even manipulates the immune system. All of these mechanisms together make treatment very difficult, not just because tumors grow, but because they dynamically resist intervention. Um, and this becomes especially problematic when we think about metastasis. So tumor cells don't stay local, they enter the vascular system and spread to distant sites. So now we're no longer dealing with a localized disease, but also a systemic one. And once the disease is systemic, local treatments alone are often not enough, and we need systemic control. And even though we do have a wide range of therapies, for example, chemotherapy, radiotherapy, immunotherapy, targeted therapies, most of them still struggle to reliably control systemic disease. They are effective, or they can be effective locally or in specific patients, but they're often invasive and not very consistently, they don't consistently reproduce across individuals. So the key issue here is not a lack of treatment, it's a lack of predictable system level control. If we zoom in a bit, two of the most interesting approaches are immunotherapy and radiotherapy. And individually, they can both work. But what's really interesting is that when combined, they can improve response rates and in some cases, increase overall survival compared to either treatment alone. And this brings us to the phenomenon I'm focusing on, which it's called the abscopal effect, which come from two Latin words called ab, which means far away, and scopus, that means target. So overall, it means effect far away from the target. So it's a phenomenon where local radiation doesn't just ring the treated tumor, but it actually triggers a systemic immune response that affects the distant untreated tumors. So what happens mechanistically is that radiation causes tumor cell death, it releases antigen, it activates dendritic cells, and primes T cells in the lymph node, which then circulate and attack tumors elsewhere. So in a sense, the immune system turns a local treatment into a systemic one, giving us a kind of treat one, kill all, or kill many approach. But there is a problem. The This effect is extremely rare. There are decades of reports, both clinical and preclinical, but it happens inconsistently, unpredictably, and in very specific conditions. So the question is not can it happen? We know it can. The question is why does it almost always fail? And that leads to the central question of my work, which is can we transform these rare, almost accidental in some way, systemic responses into a reproducible, controllable immune state? So if we think about where this systemic response is actually orchestrated, it really comes down to the lymph node, which is the bridge between the local and the systemic response, um, which means that it is not just a passive site, but also a regulated decision-making environment. Because there are, for example, threshold effects, meaning small changes don't always produce small outcomes. There are feedback loops that can amplify or suppress activation. And importantly, also, there are a lot of bottlenecks like antigen presenting cell migration, T cell priming, and exit, that they essentially filter which response propagate. And all of this is happening within a structured spatial environment that determines where interaction happens, who meets whom, and when. And this is the actual determinant of the outcome. So when we zoom out, systemic immune control is really a sequential process. So we have initiation on the left, priming in the lymph node in the center column, and then deployment to the tumor, which is the right column. And what's important is that each of these stages introduces its own constraints and bottlenecks. So failure is not just about one mechanism going wrong, it's about failure to successfully propagate through this entire sequence. Um, but we have a problem in the sense that when we try to study these experimentally, we typically rely on current approaches, which are, for example, are 2D cell culture, 3D tissue models, organ chips, animal models, omics imaging, computational models. And each of these give us very valuable information, but only a partial view. So they either lack spatial resolution, temporal control, human relevance, or also the ability to systemically perturb the system. So we can observe pieces of the process, but we struggle to capture the full dynamic system level behavior that is necessary to understand and leverage the abscopal effect. So putting this together, just to recap, we have a very complex model. We have a treatment that are efficient locally but struggle to produce systemic immune control. We also have rare but powerful systemic responses like the abscopal effect that we yet barely understand and thus can leverage. We have the lymph node as a bridge between local and systemic responses. Um, and we also have a structural bottleneck and spatial constraints that govern outcomes, but we currently lack a framework that can connect all of these pieces in a way that is predictable and controllable. So what we need is a platform that can actually close this loop. So for a system that, for example, we can measure, which we can perturb, and maybe steer immune trajectories. What this means is that we want to not just observe what happens, but actively understand how to drive the system towards the desired outcomes. And importantly, do this in a way that is human relevant. So stepping back, this is the long-term vision. Instead of running isolated experiments or building static models, we want an adaptive system. On the one side, we have the physical platform like organ on a chip or multi-organ on a chip in this case, where we observe the real immune dynamics. And on the other side, on the right, we have the digital twin, which is what I'm going to talk about today, which is a mechanistic model that learns from that data. And the key is that they continuously inform each other. Experiments update the model, and then the model suggests new experiments and intervention. So instead of just observing biology, the goal will be to move towards actively learning and controlling it over time. And again, still being grounded in human biology, which is very important. Again, but in this work, I'm focusing on a specific part of this pipeline, specifically in the mechanistic modeling and so the first is the mechanistic modeling, building a spatially and temporal explicit model of the system, in this case, a lymph node. and then the short modeling, so we can approximate this complex simulation and explore the system more efficiently. So for those of you who do not know what a mechanistic model is, at a high level, mechanistic models try to encode the actual rules that generate system behavior. So they just not feed patterns, but try to represent cause and effect as well. And there are kind of three ways people do this. There's more, but these are the basics. On the continuous side, we have ODs and PDs, which are great because they're simple and fast, but they average everything out. And the problem is we lose individual cells and interactions. Then the stochastic models add noise and variability, which is more realistic, but still mostly a population level. And then on the right, we can see the discrete ones. Then we have agent-based models, where we explicitly simulate individual cells and their interactions. And that's what we focus on today, because that's where heterogeneity and emergence actually comes from. But of course, it's computationally heavier, and we'll see later on that can actually also be a problem. One way I like to think about agent-based models, if you're not familiar with it, is kind of like a chess game. So each piece or each agent follows simple rules locally, but when we put them together, we get a really complex system level behavior. For example, a pawn, if we look at the chessboard, a pawn can only move one step forward. That would be a movement rule, but it can only interact or kill diagonally. That's an interaction rule. So now no single pawn obviously is trying to win the game, but when we put it all together, each following simple local rules, we get that strategic behavior at the system level. We can also think about it at the population level, which is also easier to understand, where agents can be humans, for example, that we interact with the environment, and we have the different states as well. Like, for example, in this example, a child dating childbearing or senior. If we put it in academia, for example, it could be undergraduate, master's, PhD, postdoc, PI, for example. But in our case, the agents are cells, like T cells or dendritic cells, and their environment is the lymph node. And each cell moves, interacts, and makes decisions, and from that, global immune behaviors emerge. So instead of prescribing outcomes, what we try is to let them emerge from interactions. Now, so we just saw that a key part of the agent-based model is the environment. However, in many existing AVMs, the environment is simplified as an empty space, for example, a 3D empty cube, where agents can move freely. But for the lymph node, we thought that this assumption wouldn't hold. Another, because, for example, the FRC, or the fibroblastical reticular cell, constrain how cell moves and how they can interact. Another common approach that people uses is to use random networks, but again, the FFC network is not random. It has a lot of structure with specific connectivity, clustering, and even small world properties, and that matters because it determines who can meet whom and how fast. So instead of approximating it, we explicitly reconstructed the FRC network inside of our simulation, as you can see on the top right. And we also structure the environment into biologically relevant subcompartments, including regions like the subscapular sinus, the paracortex, the medulla, and entry and exit zones. You probably won't be able to see much because the majority is it's green, it's the paracortex, but on top you might see some red dots, on the bottom some blue dots. Those are the subcompartments. And then we validated that structure against experimental data, matching properties like degree, clustering, small world behavior, as well as the distribution of radius, porosity, and stiffness. On top of the physical structure as well, we also modeled the molecular fields, like, for example, the chemokines, and we have the different chemokines and different cytokines. But to make this simple, these spatial gradients guide how cells move. So mathematically, the gradient just tells us the direction of steepest increase. So in other words, this is which way a cell should move if it's following signal. Then we use the Laplacian, which captures how concentration spreads out over space, which I like to think it as measuring how a point differs from its neighbor, driving diffusion. And finally, we also include decay, so signals don't accumulate indefinitely, and they diffuse and degrade over time. So together, these define how signals propagate through space and time, and this is what drives chemotaxis. So cells moving toward or away from signal. So now, after these molecular fields and the previous slide with the FRC network, we have two layers for the environment, which is structure shaped the movement and signals guiding it. So as an overview, before talking about the agents, this is a full framework. We model how, for example, antigen-bearing dendritic cells enter the lymph node, how T cells enter through the HEVs, which are on opposite sides of the lymph node, and how they migrate through this structured environment until they find each other. Then critically also we model obviously their interaction with drive activation. So everything is happening in space over time under constraints as well. This slide just makes the logic more explicit. For example, here we have our two agents, which is the dendritic cells on the left and T cells on the right. So on the dendritic cell side, we have antigen availability, maturation, and licensing. On the T cell side, we have migration, contact, signal integration, and activation thresholds. What's important is that activation isn't guaranteed. It depends on timing, location, and whether productive contacts actually happen. So this is where those gates start to merge with access, timing, and competition. Here we zoom into the agents themselves. In our case, we have two. We have dendritic cells and we have T cells. Dendritic cells go through licensing stages from antigen loaded to fully mature, and IL-12 producing cells. T cells, in contrast, go through activation states from naive to activated and eventually to effector or memory. And once a T cell is activated, it undergoes clonal expansion. So we don't just track populations, we track individual cells and their lineage over time. And then we can trace outcomes back to the original cell. Here on the right is an example simulation showing these dynamics in a very small cross section. If we see these lines over here, they're a little in gray. This is the FRC network, and we have, let's see, yes. If it works, we see that cells do follow this network and they do not go freely. We also see the red dots, which are the cells coming from one side, and the blue cells coming from the other one. And if we take a look at the scoreboard at the top left, as time goes up, more cells enter, and there's also the switch on states, and then they go back down because that means it left, basically. So now to demonstrate the model, we designed two case studies. For the first one, we asked a simple question: how does stromal topology, basically the structure of the environment, affect early T cell priming? And importantly, how much does it matter to model it correctly, since most models miss it? And to answer this, we compare different types of networks. Perfectly structured lattice, which is B, which is not biologically realistic, but a lot of models use. A more realistically structured network, which is the one I just presented 2 slides back, and this with and without Kikimokai guidance, and then an unstructured network, which is number C, well, letter C. We get biology across all the same, and the only thing we change is the spatial organization. Looking at the correlation score as well, we can also see the difference between the structure tested in the perfect lattice. These things are highly correlated. In the structural network, the correlation is lower, but still present. And in the structural case, it essentially disappears. And we can also see this in the edge length distribution, where the structural network has a constrained geometry, while the unstructured one is much more spread out. And when we actually run the simulation, we do see that structure matters. The structure environments gives you more activation, especially early on, which already tells us that the structural architecture is shaping who gets into productive encounters during that first scanning phase. And I think the most important point here is how it matters. Because if you look at the conditional timing curve, once a cell actually gets into a productive interaction, things looks pretty similar across conditions. So it's not really that topology is making the actual priming process faster once contact happens, but it's acting earlier than that. So what topology is really doing is shaping the encounter landscape early on. So basically, who can find the antigen bearing the dendritic cells, how efficiently they can access them, and whether they do so during that narrow early window when priming opportunities are still available. Um, that early window does matter because these are rare cells in a crowded structure space. It's not enough to just eventually make contact at some point. Early access changes, which cells even get the opportunity to enter the response in the first place. So even if some of the difference compares later on, that early bias is still really important biologically. Um, so overall, the main takeaway here is that the lymph node architecture acts like an early temporal gate. It doesn't necessarily change the intrinsic priming program after contact, but it does shape which cells get a chance to prime and when. In the second case study, we shift from structure to competition, and we varied the three things: T cell density, crowding, and dendritic cell supply. So now the question becomes, how does the competition for limited dendritic cell affect activation? And looking at the heat map, the first thing we see is that really strong effect on exclusion. As T cells demands increases, more and more cells fail to ever form a productive contact. So they're just not delayed, they're completely excluded from the response. And even when increased DC supply, that only partially rescues it. So we still have a large fraction of cells that never access antigen at all. So already here we see that the system isn't smoothly degrading, it's filtering, some cells get in and others don't. Um, then we asked, is this happening because interaction are slower under competition? And when we look at the timing of the first production contact, conditions on success, the curves almost completely overlap. So when the cell actually gets access, the timing is basically the same, which tells us that competition is not slowing down the priming process itself, but it is acting earlier by controlling access again. So as the conclusion of the paper was that the mechanism here is fundamentally different from what we might expect. Since it's not a kinetic effect, but it's an access bottleneck. Okay, so from these case studies, we started to understand what's happening. But the real question is, can we actually open up the system and figure out where it's breaking down? Because right now, from a clinical perspective, we observe outcomes, success or failure generally, but we don't really know which step failed inside the system. It could be access, it could be signal, it could be timing, but we don't know because the system behaves like a black box. So we know somewhere inside here something is going wrong, we just don't know where. And now, so we take the advantage of the ABM we built earlier. Since it already contains all of this information, because we're tracking every interaction, every contact, every state transition. So the information is there, we just need a way to extract it. So what we do now is, instead of treating the model as one large simulation within a single output, we can reinterpret it as a sequence of mechanistic gates. And this is important because each of these gates represents a specific point where the response can either progress or fail. So first, we have the dendritic cells that have to enter, encounter antigen, and become licensed. If there aren't enough mature disease, the response can fail immediately. Then the nifty cells enter through the HEVs and scan the FRC network. The next question is, do they actually access a mature DC? After that comes the temporal gate. The T cell and the DC not only have to meet, they have to meet with the right early window. Even after activation, success still depends on spatial context, crowding, and whether the cell remains in a productive zone. And then there's a checkpoint gate where suppression can still dominate and shut the response down. And finally, there's an egress or exit gate where activated effector cells still have to leave the node to generate systemic amplification, because everything can go amazing, but if that happens in a place that is far away from the exit, it still might not get amplified. So instead of one black box outcome, we now have a series of interpretable failure points. And once we do that, the question changes. Instead of asking why was activation low, we can ask something more specific, like was the problem the early access, or was the problem the dendritic cell availability? Was it X, Y, or Z? And that's where intervention layer that I added on top becomes important, because once the system is written in terms of gates, interventions are not generic anymore. And now we can see what type of treatments can target one gate or another one. But we run also into a problem. To really understand or control this, we would have to simulate a massive number of combinations with different arrival times, different densities, different spatial structures, different intervention schedules. And with a full agent-based model, that is a little complicated. That because, for example, mine takes for one simulation takes 20 hours to run. So if we want to know everything, it would take years for it to run. So we're in a situation where we understand the mechanism, but we can fully navigate them to the extent that we would want to understand everything. So that's when we need to learn a way to learn the system's behavior without simulating every possible trajectory. And that's when we turn into surrogate modeling. So very briefly, moving into surrogate modeling, the question is how do we choose which simulation to run? Because every simulation is expensive, so we want each one to give us as much information as possible. A simple approach would be random sampling, for example, but the problem is that random sampling tends to leave gaps in the space. Another one, which is very commonly used, are structural designs like Latin hypercube sampling, which ensures that we cover the space more uniformly. But even that is not enough, because what we really want is not just good coverage, but we want to adapt based on what we've learned already. So instead of sampling everything up front, we iteratively fit a surrogate model, identify where we are uncertain, and choose the next simulation accordingly. And this is what we call the sequential design or active learning. Now, in our case, the problem becomes even more challenging because the behavior we care about, which is the synchronized immune response, is actually a rare event. So instead of modeling an average response, we define a score that captures when the system enters this synchronized state. And when we map that out, we see something really important. These rare synchronization events don't happen everywhere. They exist in very narrow regions of parameter space. So there's no simple monotonic trend. It's pretty messy. It's non-linear and highly localized as well. So on the right, when we look at joint interactions, we start to see these small pockets where synchronization appears. So the takeaway is this is not an optimization problem over a smooth landscape, but instead a search problem over a sparse structure space. And if we sample naively, we will almost never hit these regions, which is why the design strategy becomes everything and why we're spending so much time on this as well. So now we are bringing in surrogate modeling, specifically the Gaussian processes, to guide where we sample next. And the key idea is we use an acquisition function to decide where to go. And there are a few standard strategies. For example, the active learning McKay or ALM, which samples where the model is most uncertain locally. Then there's the active learning cone, which ALC, which samples where you reduce uncertainty globally, as well as the feature information on the right, which samples where you learn the most about the model parameters. So each of these is trying to improve the model, but they're all doing it from slightly different perspectives. And importantly, none of them explicitly cares about the rare events. Um, when we apply these methods to our system, we can reconstruct the response surface, and you can see both the mean predictions and then certainty estimates, both for the ALM and ALC here. And they do a decent job of learning the general structure, but here's the issue: they spread their efforts across space. So because they're trying to model everything, which means they're not specially targeting the regions where synchronization actually happens. Um, Same with feature information, although it behaves a bit differently, since it focuses more on learning the underlying model parameters. So we get a different structure in the surrogate, but again, the same limitation applies. It's optimizing for model quality, not for event discovery. So even though the surrogate improves, it's still not guaranteed to find the rare regime efficiently. And we can see that clearly here as well. On the left, overall prediction error improves for all methods in comparison to LHS, which is the green line at the top. So from a standard modeling perspective, everything looks fine. But on the right, when we care about actually capturing rare events, the difference becomes much more important. So standard methods, as we can see, for example, if we look at the green line, it's the one out of the three methods that performs the worst with out of sample root mean squared error, but it performs the best on the right graph. So, but overall, standard methods miss parts of the tail under sample and high synchronization regions, and struggle to represent the rare behavior accurately. So, optimizing for global accuracy is not enough. So that's why we introduce a rare event targeted sequential allocation, or we call it RETA. So instead of using just uncertainty, we explicitly incorporate the probability of entering a rare event. Um, the acquisition function becomes then uncertainty times the probability of exceeding a threshold. So now we're not just asking where is the model uncertain, but we're also asking where is the model uncertain and likely to contain something important. I've talked a little faster than I thought, but I put a couple slides for current future work that I thought I didn't have time to go over. Just to briefly zoom out, I want to give a quick overview of some of these additional directions we're working on. I'm happy to discuss any of these in more detail afterwards if anyone is interested. Um, but on the mechanistic side, beyond targeting individual gates with specific interventions, one direction we're actively working on is building a more integrated multi-component model. So instead of modeling the lymph node in isolation, we explicitly connect it to the tumor site, for example, skin treatment with radiotherapy, and then track how signals propagate between compartments. And at the same time, we're working toward integrating high-resolution experimental data sets, like spatiotemporal atlases, to better ground these models, in this case, in the skin one, in biological structure. And we're very lucky to be collaborating with Dr. Wapi from Colombia that has an amazing 4D multimodal atlas. On the surrogate modeling side, we want to go deeper into the Reddit framework and convert it into the object-driven acquisition strategy. Here I put 3 formulas, but we have a lot of different, around 50 different formulas that we want to try that goes from plausibility to boundary to severity to risk and then information decision. We also want to try the deep learning to give us the ability to scale to much higher dimensional space and capture complex nonlinear structures in the system. But the limitation there is that we probably lose the calibrated uncertainty. One direction we're really interested in is the deep Gaussian processes and learning. And finally, in the control side, we have a model that's we're starting to ask what is the optimal intervention strategy over time. And so here the system evolves. So our goal would be the to be able to turn the action space becoming combinatorial and time dependent, for example. And the goal would be to learn a policy that maximizes some notion or benefit, like immune activation or tumor clearance, while penalizing toxicity as well as treatment burden. So instead of optimizing a single endpoint, we're explicitly balancing trade-offs, and hopefully we would do that with different uh, radiation doses, fractionation schedules, sting activation, checkpoint blockade, etc. So and that's what starts to connect this to all this work to patient-specific planning, where treatment is no longer fixed but adapted based on system dynamics. Um, Thank you.
Thank you so much. So we have some time for questions. If you would like to come off mute and ask your question, you're welcome to do that. If you would rather type a question in the chat, I can ask it for you. Either one works. And I can get us started here. So I was wondering if you could talk a little bit more about defining your agents as cells and, for example, like how many rules does it take to define a T cell versus a dendritic cell? Is there any overlap where what are you pulling from to come up with like the roles or identities of your agents?
Yes. So most of the rules are based on behaviors that I already know. So from the literature, and they're already pretty separated both in structure and in their behavior. They even come from different parts of the lymph node. So they normally do not even interact until well, the goal is for them to interact. But then you in terms of the rules, it depends also on the states. They can be some people do naive and activated, and that's it. We try to also incorporate naive, activated, but then all the blast and subsequent cell differentiation to be able to see if once it got activated and blasted, does it go to exhausted, to memory, um, or not nephron. So this is all, yes, that's what we're trying to do, and hopefully down the line we also are gonna do some studies that will look at outputs and see which T cell is what was coming from.
Thank you.
Thank you. Great talk. I have a question. Early on, you mentioned about the agent-based models. They are computationally heavy, and each simulation takes about 20 hours. What computational infrastructure are you using for this? Like especially when you say you do the parameter sweeps, um, like are you running this on the HPC cluster, or is there some kind of parallelization? How are you handling the computational load?
Yes, yes. So I'm using the Nagishi cluster. Um, so I'm using that. I also do parallelizations, but it's still taking a lot of time. So that's why we try to see if we would get more computation or more computation power. But then we decided that the best way, especially if now we couple the lymph node, okay, so which is one agent-based model, to then two skin ones, to have a three compartment agent-based model. That would be a little too much, and we switch to the surrogate modeling.
Thank you.
I guess sort of in a similar vein, you talked about how complicated the environment is that you're defining because you're trying
Mhm.
to use the lymph node as an environment instead of sort of a more blank scape. Is that part of what's driving the computational load is the extent of the environment. Does that make more of a difference than like how many um, agents or how many like different other parameters you're running?
That's that's a good question. I haven't tried to run everything without the environment.
Mhm.
Well, once, and that, well, apart from in the case study, and it is true that, and I've never thought about it, but it is true that the simulation did in run much faster on the ones that it was just the and the cube alone, and even if the lattice was um, perfectly structured. And the ones that took more time were the ones that were unstructured, and then specifically the ones that had our FRC completely. And even if when we added the actual chemokines on top, um, I didn't actually take a look at that. But no, it's true. No, it's a great point. But yes, I think that is one of the main causes. And also, we're even trying to incorporate more things in the environment. So it's definitely something we need to take into account and see if it's worth it, and see how that would exponentially increase the computing time.
I have another question. Your visa acquisition function, it combines the model uncertainty with the rare event. So how sensitive are these parameters for, yeah, for the thresholds that you set?
So your question is how sensitive were the parameters for?
Yeah. So the threshold for defining the rare event,
Mhm.
like how sensitive are those?
They're quite sensitive. They're quite sensitive. We'd have problems with that a little bit. Um, but that's why we have on the future steps, I put 3 new formulas, but also we have 50 more, and we want to try and see how those work, and see if it's better than the current Reta one, or if it's still, because Reta did make it better a bit, but it was not a miraculous solution. So that is something that we need to take into account as well.
Um, yeah, it looks like there's a question in the chat.
Yeah, we have a question in the chat, which I don't think is naive at all. So the question is, how does one validate the physiological relevance of your models? And can the effect of conditions such as infection be tested on your model?
Yeah. So when you say for the first question, when you say the physiological relevance, is that like how do we validate it experimentally, or how do we know the model is important?
Um, I think maybe a little bit of both, but I was interpreting it more as how do you validate
Okay.
it experimentally?
Okay, yes. Yeah, so yeah, that's a great question. So one of the things of mechanistic models is that they need to be grounded in experimental data, or the other way, and they have to be fit at the end to actual results. So for this one, we had some data that we used to to develop it, and to calibrate it, in terms of how we kind of validate it. We did some results and some calibrations, and they did all match the what's it called, our results. However, we also are planning to do some experiments on an organ on a chip, since we're half computational, half experimental, and we are planning to fully validate this afterwards. But for now, we just fit it and see if the trends matched and the some other published results. And in terms of the second one, was it conditions such as an infection? Yes, I mean, we're not working with that. We're working mostly on cancer. However, yes, 100%. I know there's a PI in BME, Dr. PNR, that is working on that as well. Is working on specific infections and the epidemiology. But yeah, we could definitely switch our goals, especially since we only have the lymph node now. I think it would be easier to switch to another use case, and then obviously the rules right now will probably stay pretty much the same since it's only from the time that the DC enters the lymph node as well as the T cell enters the lymph node, and these should be quite universal. There is true that the environment would change. It depends on what's happening, and the signals and the co-stimulation. But that is definitely something that wouldn't have to be changed as well. But yes, it could be applied if some of the rules are changed.
Thank you. Okay. If anyone has anything else, please write it in the chat and we can hang out another minute or so. Um, and I'll see if anything comes up. But otherwise, thank you so much, Laia, for sharing your work with us today. I think you did a great job, especially of sort of walking through what is quite a complicated system that maybe a lot of people have varied backgrounds in trying to understand models of this type, and sort of walking us through that and having really helpful visuals as well. So thank you.
Thank you so much.
Okay. Have a lovely Friday, everyone. Bye.
Bye.